I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
What difficult problem in oncology do you think AI is going to actually help solve?
It's a good question. Maybe faster molecule discovery, faster go-to-market.
Probably like specialization to like the patients and whatnot.
I think right now AI is pretty good about helping with brainstorming, like new ideas that you may not have thought about, as well as it helps with the huge amount of data that we have today to kind of make sense of it, right?
It would probably be something like finding new targets.
Early detection, perhaps, but I'm only guessing.
Wow, that's a very good question. I think there's really like a lot of really good ideas and thoughts that we can think of. For example, like you can ask AI to do literature search and go around the clinical trials and design like even like the different trials in terms of like how I can like drive my clinical trial forward, and then with the specific drug that I designed for a specific population of patients, like how can we expand that to other populations with the interest of their genetic background, and then all of those like big data that's embedded. I'm excited about it.
Hi everyone, welcome to Two Scientists Walk Into a Bar. I'm your host, Danielle Mandikian. Over the last 7 seasons, oncology episodes have definitely been some of the most popular. And it's not just the prevalence of the indication, but the real breakthroughs that have kept our hopes and curiosity peaked. So we are so excited to invite Anwesha Dey to the bar to help us learn more about AI and how this is revolutionizing the field of oncology. Specifically, she utilizes AI for novel target discovery. Anwesha, thank you so much for joining us.
Thank you for inviting me. Pleasure to be here.
So to kick us off, I'm going to ask you to take us back in time. What was your journey into research pre-AI?
I would say my first exposure to research was as an undergraduate student, and I absolutely fell in love with research. My PhD was in structural biology in NMR spectroscopy, and as a postdoc, I made the switch into cancer research, mouse models of cancers, and then started my lab in discovery oncology, where I got to work on signaling pathways and nodes that are important and deregulated in cancers, and then eventually really fascinated by the drug discovery process and how to translate those ideas in the lab, the basic research ideas, and results into making medicines for patients.
When we look at the oncology field, what do you think are some of the biggest breakthroughs for oncology therapies?
If you ask me to name one, I would have to say KRAS, right? Being able to target KRAS, what we all once thought was undruggable, right, as an oncoprotein. Knowing the structure, finding that pocket, doing that chemistry, and finding that molecule. That really opened up the field. But the thing beyond that is this was so fundamentally important because that one study, which started from KRAS G12C, opened up the field broadly for KRAS into the other mutants, right? From KRAS G12C to G12D to now so many others. But this also brings up another aspect there on what might be some of these emerging drug discovery technologies that make these possible. There, right now, it seems like broadly for the field as a whole, there's just so much excitement about molecular glues because they could be the next, or they are in some ways the next set of as tools, technologies, as we understand more about the biology. It's a great way to then bring together fields of drug discovery and basic biology to really uncover the rules of the game, to make the next set of targets, the undruggables, tractable.
Hi, Danielle.
Hey, Wellington.
Hey, Danielle.
Hey, Karen.
I know this has come up in other podcasts, but can you tell us what KRAS is again? And why is it so hard to target?
Yeah, KRAS is just an acronym for this really important oncoprotein. It's a signaling protein that's really critical in a cascade for development of cancer. And a lot of people were trying to target it, but it was really actually hard to get something that bound to it well, just because people didn't even know the structure of it. So that was one of the big breakthroughs in the field, is having these crystal structures. It gave people some kind of ledge to build off of for binding. And whenever she was talking about the different variations of like D, C, whatever, these are just different mutations that have been found on that protein.
Danielle, my first question is about molecular glues. What are they?
Whoa, Karen, get out of my brain. I was just about to ask her that question. And for our listeners, can you explain what molecular glues are?
So you can think of molecular glues very simply as a glue or a small entity that glues your target of interest to perhaps the cellular degradation machinery or another molecule that takes it to the cellular trash to get rid of it. The field is emerging in so many ways that it doesn't always have to even degrade. It could sequester your target, right? So think of it as a way to glue two aspects that you want to bring together to go after.
This is horrible, but that's actually how I kill spiders. I have a huge fear of spiders. Yeah. Okay. So you named KRAS as being like one of your favorite breakthroughs. What's something that maybe hasn't gotten there yet, but you really think is going to be super promising, that you're really anticipating as being like a big showstopper at some point?
So if you think of KRAS, right, as most prevalent oncogene, or the flip side of oncogenes are tumor suppressors. Now that's an open field in some ways. What's our guardian of the genome?
Right.
P53.
Yeah.
Right? So it feels like that is the next, in some ways, wouldn't that be an exciting next frontier? There's a lot of work going on broadly across the research community. We've been working on this for so many years. Scientists have been, like, I guess if you PubMed the number of papers on P53, it's a staggering, shocking number. We still don't have a way to make that druggable. So that is something on the biology front, I think. And not just biology, because that's a clear example. The tumor suppressors are lost, right? And the fact that we don't have one for all the P53 mutations or loss of P53 is not for lack of trying, right? Mm-hmm. So this is an example where, together with the biology, it's going to be a really strong collaboration with drug discovery, right? The modalities.
Yeah.
And so I feel like the tumor suppressors is one where we can truly bring together biology, drug discovery, new modalities. And that is one that I really hope is the next KRAS frontier of sorts, would be tumor suppressors. And I said I'd give you another one, which is also a super exciting one, and that's in the context also with AI. Mm-hmm. And I would say AI protein design, mini binders, and really thinking of how can we leverage that. Mm-hmm. And the field is so rapidly advancing in the context of mini binders, AI protein design, lots of labs, lots of companies. The next step is really making that into real medicines, right?
Yeah.
And so a really potent binder is not the same as a medicine.
Danielle, definition alert. What is P53? It sounds like an airplane.
P53 is like one of the most classic proteins that people have been interested in oncology. It's really an essential protein in understanding within the cell when there's a problem and knowing to kind of when to self-destruct. And so when there's a problem with P53, cells don't realize that things are messed up and they just keep proliferating.
Anwesha was talking about AI protein design and mini binders at the end there. Can you explain a little bit more about what those are?
From the way I understand it, they're using AI to figure out, hey, if I have target X, I need this protein structure in order to bind it. And in the context of mini binders, like instead of having to design a really large biologic like an antibody, you can design a much smaller protein that contains that binding domain, and that's what they're referring to as a mini binder.
And do you think that whole field is like AI protein design?
Mm-hmm. Well, I mean, like AI protein design is really just saying, like, if I want to have a structure that is doing something like binding to a target, what would that protein have to be to have that structure? Because like one of the things that's really tricky with proteins, and I think like we had Rich Bono kind of talk about that a little bit in a previous episode, but like, you know, when we think about protein binding, proteins are not just like a linear chain of peptides. They have these 3D structures within themselves and with other proteins that form like a larger complex. And so if you need to bind to something, it's not as simple as saying, hey, I got to have these amino acid residues that have these properties that are going to bind. You have to figure out what that whole 3D structure would be in order to have those amino acids in the right, you know, 3D space to actually bind to that target. And so that's like a whole thing. Recently in the literature, there's been a huge uptick in how clinicians are utilizing AI for diagnosis. Are we seeing that same kind of momentum in early research?
Absolutely. If you think about that, say early research, the biggest momentum is in that drug discovery, the molecule making phase. And the next one that is also equally exciting, but a lot more to do, is in the target discovery phase. How are we leveraging AI for finding new targets from all of these large data sets? We have heard about the virtual cell and the foundation models. You've had podcasts on this before. So yes, absolutely. Early research is super exciting.
So I've heard you suggest that we should really be thinking about AI as a member of our team. Can you go a little bit deeper and help us understand how is that going to push oncology forward?
So as I think of our new team member as AI broadly, it's really many members under that same umbrella, one member. So if you think of that umbrella, one member as our AI agent, the first one, you have a tool, or you think of an AI agent, which is really good for literature. You're getting into a new topic, right? And you want to know all about it. Now, certainly you cannot read 30,000 papers right, within a reasonable time. But there is a great agent. You have a great team member who will give you a fabulous synthesis. You ask the right questions, you put in the right prompts, and that almost needs a masterclass in and of itself. Like if you're really good at prompt engineering, really know how to ask the right questions, right? You have an agent for literature synthesis, right? Then there is one, you are brainstorming. You need new hypotheses, right? And you have one team member doing that. You are trying to understand modality. You have one agent thinking of that. You did some experiments, you have a bunch of data. You have one agent who's doing your data analysis. Based on this analysis, what should be your next experiment? And you have ideas brainstorming on that, right? Then you have another aspect of, I did these experiments, I got this data, it got me thinking about I should have some more new ideas, some more new hypotheses. What would those be, right? Think of all of these coming together. And you're doing this with your teams as well, right? And everybody has all of this. Like think of the opportunity there to truly augment and accelerate you. And then you're done, you go home at night, right? But before you go, while you're sleeping, before that you had uploaded some of the data sets, put in some basic question, and you have an AI agent who, while you were sleeping, looked at the data sets, went through a bunch of those data sets, came up with some hypotheses, tested some hypotheses, and did some analysis, ran some more additional analysis based on the results, and then morning came up with, these were the hypotheses. True, maybe not true, tested them based on your data, said this is true, likely true, some of them were wrong. And next morning you come in, you have all of this, right? But then it's not like these are all perfect. This is not all solved. And this is not a replacement of your scientific judgment. What I find is really exciting is that there is truly an opportunity to do this at scale for all of us to do that, right? And think of it as a way to scale and augment what we're doing. Now, is this all perfect? And is this that we don't have anything to do? No, not at all. Because there are some absolute critical watchouts. And then if we discuss some of those, on the literature search, the thing that is emerging often across, if you evaluate a lot of these, is that many of them, or some of them maybe, are very, very good at deep, narrow, specialized tasks.
Okay.
Right? So I have an agent that does really good literature synthesis. But that doesn't mean that makes it great for hypothesis generation. That's a different one, right? So you have to know which of your agents is really good at which task?
Like, what's the scope for each one?
What's the scope for
Yeah.
each? Within those scopes, the thing that becomes absolutely critical, and that sort of reminds me of, how when we were growing up and you go to do math, or now we do that with our kids, show your work.
Yeah.
I'm not just interested in your math answer. I also want to see your process of thinking.
Yeah.
It's like trust but verify. It's not that you come in and you see an output, a polished report is not a reliable scientific report.
Totally. 100%.
Right?
Yeah.
So these are the things to keep in mind. Just because you had an excellent report next morning or an excellent analysis done next morning doesn't mean that it's correct. That's where we all come in. That's our scientific judgment.
Yeah.
Some agents, they show their work, including the code, right? That's where at the end of the day, things that are auditable, right, reliable, trustworthy, and at the end of the day show their work, right?
Yeah.
Those are the ones over time you get more confident in using, and they're all getting better.
Danielle, so much talk about agents that I feel like I need to get one. But in all honesty, though, there are other tools that we're using in our AI toolbox, right, for AI or for oncology research. And what are they? Can you remind us?
Yeah, we've totally touched on this across different episodes, and it's actually helped me learn a lot from, my vantage point, right? And so there are large language models that are helping us kind of translate the language of proteins. There's foundational models that I can't even remember the specific examples because those are just so massive in terms of like answering really complex questions. But we're putting all these different kind of biology information into these foundational models so that we can ask more complicated questions about how proteins are actually interacting with each other and what could be the consequences of this. And so whether we're looking at a disease indication like, I don't know, like, some kind of like immunology response to something, or we're looking in the oncology space, all of it is really centered around understanding complex systems and what the biology interface can actually be used to intervene with.
Are you a secret agent?
I would never tell. Could you give us some examples of how you're actually directly applying this within the oncology space?
So I guess one way to think about it is this is such a rapidly evolving field. As things are progressing, agents are evolving, our understanding of how to use them is evolving, how to ask the right questions, the right prompts, and how to match the right questions in oncology with the right agent is evolving. I often think of where we are really now is evaluating them. Evaluating the agents to match the right agent to the right question. So for example, right, there are certain agents which are really good at literature synthesis. I still think of them in that productivity gain bucket that we were talking about. When I'm using agents there for a new area in oncology, a new target in oncology, or should we be going after this target? What is known in the literature about it, right? What are the emerging mechanisms of resistance? What are some of the other therapeutic opportunities in this indication? So there's many questions there. When I evaluate there is which is the agent that gave me the best output. It's concise, it answered my questions, it stayed on track. Sometimes we also know, right, it's my thought partner when I'm brainstorming. Another example where I think is a good opportunity for us often, even on teams, and this is true for not just oncology, but even broadly science and research, right? We're asking a question. I already know a bunch of things that didn't work as part of the experiments that we did in the lab, right? And so then as part of the prompt, I'm like, exclude this ligase, exclude this, right? It could be any of that. Agents that stay on track and don't get completely derailed. So that's super useful, but that's also part of our evaluating the right narrow space. The other one that we are also evaluating, which I feel is super exciting, is that idea of new hypothesis generation. Right?
Tell me all about it.
So I think that's something that is really in my mind, I feel is quite in that early space, super exciting. The possibilities are endless, but real watch out, because then are we really going and doing a bunch of experiments downstream to evaluate these, right? Because that's the bucket of that creativity gain, which I find super fascinating. That's my brainstorm partner. That's where I cannot analyze all our RNA-seq data, right? I can or go through all the supplementary figures tables, all the supplementary tables of all the papers that are there on my favorite target in oncology.
I think like most of us, we're using AI more and more. Like for myself, I've used it for, harder math problems, figuring out ways to do data analysis that I haven't done before, but I haven't used it for hypothesis generation. Can you give us an example of one of these agents for hypothesis generation?
So as part of the prompt, right, what has been fun, there's some agents where you can also go back and forth, right? I put in my question, I'm thinking of this gene X. Is there something known about this before? How do I target it? What might be the resistance mechanism? What is the downstream consequence of this? Or as part of the mechanism of action, I suspect there might be an E3 ligase involved. Which one might that be? However that is. Whatever your question might be, as a response, you might get another set of, are you thinking of this? And then you have an opportunity to correct your prompt, or you might clarify, right? So the ones which are your best thought partner are the ones that you can go back and forth in. And what am I really looking for there? Often in this specific context, I'm looking for those unexpected connections. For all of us, right, for our projects, we've read a ton of papers, we've thought about this a lot, we've done a ton of experiments. But what are those unexpected connections that I didn't make, that I hadn't thought about, right? This is an opportunity to do that. Right?
I see. So it might be like, I'm interested in targeting protein X, and you go back and forth and it says, oh, hey, watch out, there's been something about toxicity or some downstream effect.
You can have it way more concrete.
Yeah.
That's an example, and you can also have it way more concrete than that. You can say, can you design some aspects of this, right? And you can go or come up with specific experiments within the realm of, this is the specific modality I'm thinking about, what is an experimental workflow, right? And what would that look like? And maybe aspects of this may not be new, but it's something that you have not yet thought about. Or you upload, a bunch of your large data that you collected over time and ask certain questions, and you might get an unexpected connection of, in this data set, it connected multiple of those and uncovered perhaps either a downstream target or an upstream target or a node that you hadn't thought about, right? These are the possibilities.
Whenever you're thinking about AI in oncology, what do you see as like the quick to address like now versus like in the next 5 years?
So one of the things that's really exciting even now and here to help accelerate the discovery process is for the targets that we have, leveraging AI for molecule making, right? Or leveraging AI to execute on those. The next phase is from all of the developments currently across our screens, foundation models, virtual cells, is understanding causal biology to then leveraging AI to find new targets. That is something that we're all headed towards. Something that will be really exciting as the next phase to see evidence, and eventually further down, all of this being an integral part of all our workflows as autonomous.
That's really exciting. Do you see this being applied more for very specific oncology indications or more across the field broadly?
Overall, where I hope we're all headed is that this is broadly applicable across all of oncology indications, right? Different people in the field, different companies are focused on different indications, some in certain areas, some others. At the end of the day, our patients are waiting. There is a sense of urgency. It's about collaborating with everybody. And I hope where we're headed is a cure eventually. Right now, we're in the process of treating. But imagine the day where we have all worked together to work towards a cure and eventually towards prevention of cancers. And AI having been an integral part of that, where we all work together as a team to make that happen.
That would be so exciting to see that really kind of explode within the oncology vaccine space. Anwesha, I really just want to thank you for joining us. This has been so fun. I always love getting a chance to interact with you and learn from you. Thank you for being here.
It's been an absolute pleasure. Thank you for having me.
Danielle, nice episode. We've done a few of these now. Correct me if I'm wrong, but I feel like that I can start to see the picture of a new type of, I can start to see science being done differently. And so this relates to the speed of how you are doing science. the fact that you may be able to do more experiments in parallel, and also this new thing that you just talked about of hypothesis generation. What do you think about that?
Yeah, everything's changing, and I think it's happening like in real time. I feel like things are changing so much more rapidly now than what I've experienced in the past. And why that is that, we're able to kind of like dig through the literature faster and more efficiently. The way we're kind of integrating the biology and chemistry is so much more massive and informative than the way it's been in the past. And then we start thinking about even like new experiments, even in my own lab, we start thinking about what do we want to do next? We have to also think about how is this going to be automated and done at scale? How do we think about this from, like instrumentation and data curation? So now we're kind of like changing how we approach things end to end. And it's really wild, but in the best possible way.
So is this unique to the oncology space, or are these AI tools being leveraged across all therapeutic areas?
We're looking at this from every possible angle. Like I work in oncology, neuro, and ophtha. And in every one of those disease indications, we're looking at how to utilize AI from end to end, just like what we've discussed in oncology. Because we're integrating all of these foundational models with such massive data sets and understanding the interconnectivity of all these signaling pathways in ways that we haven't before, all the structural chemistry for the protein interactions in a way that we haven't done before. So there's so many things that we're going to understand and discover about the basic biology that we just are going to, illuminate different things about biochemistry that we just haven't been able to approach before. What I really think is going to happen is that we're all going to go through these massive changes, and while it might be kind of like awkward as we're getting our footing, I feel like we're just starting off running with it. And what we're all excited about is how this is going to translate to patients getting medications faster, us figuring out how to approach unmet needs that we haven't been able to approach before. This is an exciting time. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stocking cells.